Hello, everyone. My name is Mclaughlin, and I have a PhD in neuroscience from the University of Pennsylvania here in wonderful Philadelphia. Hope you've had a lovely week. It's been a pretty insane and wild week. But since basically like everybody who's been even like touched social media in the past several hours and like days has been basically bombarded with COVID-19 discussion, and there's some interesting things we could talk about there. I just thought I would bring up a topic that's still related to biomedical science, of course, but a different topic. So today I wanted to talk about medicine that's one of the most commonly prescribed in the world. Even people who aren't prescribed it may have heard of it, maybe a family member has prescribed it or something like that. So I want to talk about metformin. But I don't want to talk about what it's usually used for, which is to treat type 2 diabetes. Rather, I want to talk about a study that came out pretty recently that evaluated whether metformin may have some interesting effect on slowing cognitive decline. So it was a study conducted at the Garvan Institute of Medical Research in New South Wales in Australia. So metformin, which is marketed as glucophage or glucophage, I never know how to pronounce that word, but it's just metformin. It's been used to treat diabetes for decades, and it's a pretty interesting molecule, actually. It was evidently originally derived from compounds that are found in the French lilac, which is this beautiful purplish, or I suppose it's technically lilac in color, but whatever. It was used in medieval Europe as a remedy for the intense need to urinate at the time. Which, by the way, became recognized as a sort of like hallmark symptom of what became known to be diabetes mellitus. So to be clear, some of the molecules that are present in the French lilac have chemical structural features that are like the basis or the inspiration for metformin. But anyways, ever since the 1950s or so, it's been used to treat diabetes. And today, it's basically the first line medication of choice for the treatment of type 2 diabetes. But more recently, its benefits for the treatment more recently than it was observed to be beneficial for the treatment of diabetes, metformin has been shown to possibly have some additionally interesting effects. And I say possibly because we're still not at a place where any physician would prescribe metformin for anything other than diabetes, and type 2 diabetes to be specific. So over the years, the prescription has been observed to have like interesting correlations with cardiovascular and metabolic disorders, neurodegenerative disorders, cancer, chronic inflammation, a bunch of other things too, frailty. And so, I should note that pretty much whenever you hear that any one molecule is being claimed to be beneficial for a ton of somewhat unrelated or seemingly unrelated conditions, your skepticism alarm bells should start ringing just a little bit. But that said, there's definitely a body of evidence that has accumulated over the years that indicates there could be some interestingly, just generally beneficial effects of this medication. So, the mechanism of these beneficial effects of metformin or mechanisms are still pretty controversial. There's some evidence that it may initiate a biochemical cascade that activates a process called autophagy or autophagy, depending on which side of the Atlantic you're living. So I'm not going to get into the nitty-gritty details of how it seems to activate autophagy here. But if you want some of those details, I did go into them on my podcast called Wired to be Weird, which you can see down there, in an episode devoted to longevity. We talked about more than just metformin, to be clear, but we did get into the details of metformin. The short story is that autophagy is just a sort of a broad term that means that parts of cells are dissembled and either recycled or just eliminated from the body. So another mechanism by which metformin may be beneficial for something like cancer is how it affects global DNA methylation, which essentially alters the expression of genes by either activating them or inhibiting them, making them more or less likely to be expressed. And so this property seems to be involved in how it regulates metabolism in general, but also potentially the growth of cancer. Metabolism is obviously very important for the growth of cancer. So this is still fairly new research. So these are just potential explanations for that need to be studied more widely and more widely corroborated, I should say, to be considered even close to conclusive. But they do make sense. These cascades are known to be involved in these various conditions. So all of that said, right, there's literature to indicate that there might be something interesting that metformin may be able to offer. But at the sort of simplest level, the mechanism of metformin is to just reduce the amount of glucose that the liver releases into the bloodstream, so that the insulin that's still available doesn't have to deal with like gigantic and overwhelming spikes of glucose. But research that goes back a bit has shown that metformin has effects on the lifespans of a bunch of different animals: worms, flies, mice, rats, the noble C. elegans, or nematodes. If you are on science Twitter, you might have seen the great nematode wars or C. elegans wars between a bunch of different scientists. But anyway, so like as an example, it was shown to extend the life and health span of the equivalent of like middle-aged male mice, as well as strains of mice that are prone to developing cancer. So, it's studies like that which have prompted interests in exploring whether it might have broader benefits in humans. So an example of interesting results in humans specifically is from a meta-analysis, which is sort of like a study that looks at a big collection of other studies that are asking very similar types of questions to see if there's like a broad pattern that emerges from all of those studies. And this meta-analysis showed that patients who received metformin had significantly lower all-cause mortality than non-diabetics. And then another study showed that for patients with type 2 diabetes, so these are diabetics, treatment with metformin was found to be correlated with lower incidences of cognitive impairment. And there's several other sort of similar types of studies that tend to find similar patterns, right? Patients with type 2 diabetes, when treated with metformin, tend to exhibit other benefits above and beyond what would be expected from effectively managing the diabetes. And it's worth acknowledging that not all studies come to the same conclusion, but a bunch have. And so, we can now add a new study to the bunch. So this group that did the study in Australia conducted their study over, or including a bit over 1,000 older patients without dementia who were between 70 and 90 years old at baseline. And importantly, to be included in this study, they didn't exhibit signs of dementia or any other psychiatric or neurological disease. So they performed neuropsychological tests every 2 years with these people, and the tests probed everything from, which is kind of what makes this study cool, probed everything from executive function and memory to attention, language, visual spatial function. And so they did like a similar battery of tests to identify if the participants were beginning to exhibit signs of dementia. So they measured, another thing that makes this study pretty cool is that they measured hippocampal and parahippocampal volumes using MRI at baseline and then after 2 years with about half of the participants. So not all of them, but about half. Still a lot of people. So of that large group of participants, 123 of them had diabetes, and 67 of them received metformin, while the rest didn't. So let's just, to cut to the chase here, it was participants who had both diabetes and who were treated with metformin who exhibited significantly slower declines in global cognition and executive function when compared to participants who had diabetes but were not treated with metformin. So similar findings when it came to dementia, right? Participants with diabetes who received metformin exhibited lower rates of the onset of dementia than those who had diabetes but didn't receive metformin. So, just to be clear, in case you're not exactly familiar with what diabetes is, even if you're, familiar with the word, it's one of the, most common conditions out there. There are several types of diabetes that can occur, but the one we're talking about here is specifically type 2 diabetes, which occurs when the body can't produce enough insulin to handle blood glucose levels. So as a result, glucose, which is basically like sugar or a carbohydrate, is able to sort of hang around in circulation for significantly longer time than it typically would, so long enough that it starts causing a bunch of problems in parts of the body that didn't evolve to be exposed to these high levels of glucose for prolonged periods of time. When there's all that glucose floating around in a person, they'll encounter the term hyperglycemic. So type 2 diabetes is unique in several ways relative to the other kinds, but it's most likely to occur in people who are a bit older, which is why it used to be called adult onset diabetes. But now type 2 diabetes has become quite a bit more common than it used to be, and younger people have been shown to be capable of developing it. So the name was changed. It's not just adults. Type 2 diabetes is definitely the most common form of the disease. There are about 29 million people in the United States who have this condition, and about 1.5 million Americans are diagnosed with the condition every year. And by the way, another 84 million have something considered to be like pre-diabetes, meaning that their blood glucose levels aren't exactly ideal. They're a bit too high, but not quite high enough to be considered full-blown type 2 diabetes. So pre-diabetes can convert over into type 2 diabetes if changes in diet and exercise don't happen. So of all the parts of the body that are particularly picky about what's going on in the body, the brain is almost certainly the pickiest. It essentially evolved all of these various tools to basically drive us to engage in a bunch of behaviors that make sure that caloric energy is always available to it, to make sure we don't allow it to get too cold, right, to mess up biochemical reactions that it needs to happen, to make it, feel bad, to hurt when it gets hit, so we avoid getting bonked on the head too much, and so on, right? These are all mechanisms that it evolved to drive us, or to drive us to go to sleep, at least in part, so it can do a little bit of housekeeping on the inside before the next day, cleaning up, metabolic byproducts and stuff like that. So, when it comes to a situation when there's way too much glucose floating around in the brain, there's clearly going to, that's clearly going to produce some problems because it didn't, directly regulate. It can't directly regulate how much glucose there is, right? It has to rely on the pancreas to secrete more insulin to take care of all that glucose. But when somebody has type 2 diabetes, that's exactly what's not happening. So, perhaps unsurprisingly, as we get older, people who are living with type 2 diabetes have a 60% risk of developing dementia. And the authors of this study suggest that their study has provided some promising initial evidence that metformin may protect against cognitive decline. So, it may be true that they provided evidence that this is possible, but I wouldn't say it's initial evidence or some of the earliest evidence. But it's super cool that this research group is planning to perform a randomized controlled trial of metformin in people who are at risk of developing dementia to see if, treatment with the medication has an effect on developing the condition, dementia, over the course of 3 years. Totally into it. That's a really cool study. But this has been a question on the mind of scientists for quite a while now. 2 clinical trials are devoted to exploring whether metformin might be useful for aging in general, not just for people who are diagnosed with type 2 diabetes. And I talked a little bit about them in the podcast, but they're called the TAME and MILES clinical trials. Everything in biomedical science, charming acronyms are always a central goal of your naming scheme. Except for geneticists. They come up with the most Byzantine esoteric names for genes, but whatever, that's fine. They're just the geneticists. Anyway, so the MILES trial focuses on whether metformin can restore gene expression in older people with impaired glucose tolerance. And then the TAME trial is the first randomized controlled clinical trial that's designed to explore whether metformin works just as an anti-aging tool, as a longevity tool. And so evidently the TAME trial has yet to begin. And I have to admit, the first time I read about plans for the trial, I was totally under the impression that it was already funded and they were just just working out logistics. But it looks like they're still waiting for some funding. Which, to be clear, most definitely doesn't mean that it's totally unrealistic or a bad study or anything like that. It's just that it's still waiting to happen. Clinical trials are incredibly expensive. After all, while they vary a lot depending on the type of medication that's being evaluated, I've seen ballpark figures that for a clinical trial where there's a median cost of $41,000 per patient. And, in the vast majority of cases, a ton of patients are necessary to complete a solid clinical trial, right? We're not talking about 12 people. There are some cases where that can happen, but that's sort of, they're the exception. And keep in mind, that $41,000 is a median cost. There's a paper published in JAMA. This is probably only interesting to me, but or maybe people in biomed. But there's a paper published in JAMA a few years ago, suggesting that the cost of clinical trials range between 2.1 million involving 4 patients, right? So those are the sort of more rare, small clinical trials. This was for a rare metabolic disorder, rare meaning like not that many people get it. And then on the other end is like $346.8 million for a combination of cardiovascular drugs, seeing if like a combination of these drugs would yield superior health outcomes. And keep in mind, that's just to do the study, right? There's a whole bunch of other costs that come into play. So this TAME trial is supposed to be a combination of trials at 14 different institutions, different sites throughout the United States, recruiting over 3,000 people between the ages of 65 and 79. So if you're in that window and you're interested in potentially participating in this study, maybe you can check it out. Probably the single most challenging issue for them is that, they're not trying to cure a specific disease, unless you consider, like Dr. David Sinclair from Harvard, conceives of aging as a disease. But so they're not naming a specific disease that's recognized most widely and trying to cure it, or even improve patient outcomes relative to like current treatments, right? It's just like not really how most research is funded at the moment, at least. There's only so much money to go around, and the NIH tends to prioritize disease-focused types of research, more translational research is what we call that. So the MILES trial, though, it has been completed, and I have to admit, like, it was significantly smaller than the impression I got when I was reading about it a couple years ago. And that could totally just be my fault, like just poor comprehension on my part, and I just like misread it. But it looks like they only had 8 people enrolled, one of whom ended up dropping out. So it's a pretty small group for a trial like this. And to be fair, this trial wasn't evaluating longevity specifically. They were focused on just changes in gene expression profiles in older adults who have impaired glucose tolerance. So basically they were focused on like the effects of metformin on specific biochemical pathways, not just general longevity, but the pathways they chose clearly have some at least perceived relationship with longevity or regulatory effect on longevity. So the trial did identify some changes in gene expression, but they're not really like the kinds of changes that are like directly relevant to the relationship between aging and cognitive decline specifically. And there have been other randomized studies of the effects of metformin have on the brains of people who've been diagnosed with diabetes. The authors of the study I'm bringing up today, they highlighted another study with another lovely acronym called ACCORD-MIND. And so this ACCORD-MIND trial was a randomized study that evidently showed reduced brain volume decline associated with metformin treatment, right? So theoretically it staved off declines in brain volume. So, to the credit of the study of today's paper, the authors of today's paper, they also highlight studies that have suggested no effects between metformin and cognition. So they highlight the Diabetes Prevention Program Outcomes Study. Kind of hard to remember those because there's no cute acronym. And this was like a pretty long-term study. They also highlighted that this isn't the first paper to show that metformin treatment has been associated with improvements in certain components of cognition. Like in one paper, it looked like this was even the case in people who didn't have diabetes. And so they have a nice review of like similar studies towards the end of their paper. And so if you want to check it out, I have the citation in the description below. So. All right, so let's summarize. Like, what does this study really show? Where does this leave us? For people who have type 2 diabetes, it seems that treatment with metformin may protect against some of the known cognitive impairment that's associated with diabetes. And in fact, people evaluated in the study with diabetes who were treated with metformin exhibited cognitive declines in rates of dementia that looked quite a bit like people who didn't have diabetes, even despite increased, even despite the fact that diabetes has increased cardiovascular and dementia risk factors that result from the disease. So what this does not suggest is that people who don't have diabetes, those people should not be like aggressively seeking out sources of metformin to stave off the cognitive decline that we all experience as we get older, right? It's not just people with diabetes. It's just there's varying rates of decline. And so But it is good news for people who've been diagnosed with type 2 diabetes, and there's a lot of people who have, particularly in the United States, but not just the United States. So it looks like metformin may be helpful in at least reducing some of the cognitive effects of that condition. But I think we can be pretty confident that this study will be, cited in another series of experiments published in the next few years that'll explore whether, metformin prevents cognitive decline. And so, maybe it'll be that TAME trial that I just talked about. We'll just have to keep our eyes peeled. But anyways, yeah, and if you are interested in a more mechanistic exploration of metformin, as well as like a bunch of other molecules that have been used for the promotion of longevity, like nicotinamide riboside or nicotinamide mononucleotide, pterostilbene, resveratrol, all those types of molecules, you can check out the podcast. It's called Wired to be Weird. Let me know what you think. All right, that's what I got for you today. Hey, what's up, Watchmaker? Thanks for still doing this after all these years. Yeah, well, I think if I stopped, well, at least I'm sure my wife would find me a lot more irksome. Can you talk a little bit about your live stream setup? Oh, that looks so good. Oh, thank you so much. It's pretty simple. It's just it's a webcam. It's a webcam and then some lights. You can sort of see. Lights just because there's so much light behind me. It's like so much backlighting. And I still don't really have it ideal. But, it works. You can hear me, you can see me. That's pretty much the goal. Oh, and then I use OBS, which reminds me. All right. So with that, why don't we start the live half? Beep, beep. Can we beep? There we go. Hello, everybody. My name is Ian Mclaughlin, and I have a PhD in neuroscience from the University of Pennsylvania. I think what I studied is the best way to probe free will. Kind of crazy. But what I studied was addiction. I tried, my work was to map out very evolutionarily ancient, very evolutionarily conserved, meaning a lot of animals have it, circuit in the brain called the medial lobenula IPN axis, or interpeduncular nucleus axis. And I was trying to map out the mechanistic contributions of this circuit to addiction and anxiety, I should say. And on YouTube, I just talked about a study that came out exploring whether or not metformin may have any kind of cognitive benefits, and you can check it out, youtube.com/anthropoid. All right. Oh man, gosh, I was so sure I wasn't going to forget this time. So sure. All right. Let's see, because it makes it easier to see your comments. Let's do that. Okay. So I have tinnitus or tinnitus. Can you please tell me, is there are we any closer to a cure? Yes. Yes. There was a study that came out, actually, I guess it was a couple months ago at this point, that I think was using neural progenitor cells or like cultured stem cells, something like that, for, so tinnitus can develop via a couple different mechanisms. Most commonly, it's induced by damage to the hair cells that form that enable us to hear things. They're like these little, they're called hair cells because they look like hair. It's because they're like these long, thin cells that, stick up and they vibrate at different frequencies, and they're arranged sort of like a keyboard from low to high and different notes. That's why you can tell that this is a higher note than this. And maybe you can hear my baby in the background. That hair cells enable you to do that. And I apologize on their behalf. But so usually, tinnitus will result from sound that is so high. So the volume is so great that these hair cells vibrate and then break. And unfortunately, much like most of the central nervous system, they never regenerate. And the brain receives that event as though that specific hair cell is just constantly vibrating. Its absence is sort of like it's just constantly stimulating. So as a result, you just have that long, for people who don't know what tinnitus is, it's this usually rather high-pitched sort of ringing that you're hearing, and it never goes away. And it can be quite debilitating, at a minimum extremely irritating. And so I'm pretty sure there was a study that came out that successfully regenerated hair cells. And if so, that would be the cure. Like that would be a sort of like an ideal treatment, because what's the ultimate goal here. It's to, mitigate the damage that occurred that caused tinnitus. So yes, I mean, the short story is yes, we are closer, but it's not like it's going to be in a doctor's office near you anytime super soon. But within not a crazy length of time, like maybe 10 years or something like that. Thank you very much. My pleasure. Hey, what's up, Asha? How's the little one? Yeah. Well, she so I have I keep saying 1.5 years. She's older than 1.5 years, but at this point. But that's just sort of a convenient. I just always hate it when parents say, like, you ask how old their baby is, and they tell you, like, 27 months. I'm just like, you're gonna make me do this math? Like, just tell me. Anyways, so she's quite young, still a baby, and it has been remarkable to observe all of these. Her brain is still generating new neurons. Obviously, right now her head is pretty small. Hey, what's up, Abyss? Good to see you, man. And so these circuits are beginning to sort of click on and form connections, right? The basic scaffolding is pretty much there, but different regions are beginning to get larger and become more networked. And like, she still doesn't have a particularly magnificent frontal lobe or prefrontal cortex or cortex. And so she's it's sort of just like watching a very primitive human just doing whatever they want at any moment. I'm hungry, I want this now, I have to poop, I'm gonna poop, like, and so, but what's clearly happening right now is her sort of limbic system is beginning to come online. And so she has very, very intense emotional reactions to pretty much everything. And so, which is, it's almost always, delightful, but, sometimes it's not. And so, my current strategy is when she starts, when she goes straight from, 0 to 11, 0 to 100, when she doesn't get what she wants and just starts crying, I just stop giving her attention. Right? Because, your immediate reaction as a parent, as a primate, is to resolve the suffering that you're perceiving communicated by an organism that can barely talk. Actually, she can talk. She's doing sentences now, but, relieve the crying. What's causing the crying? Let's make the crying go away. And it like her, like it has been shown that, like, babies crying is a remarkably, like, repulsive and irritating experience, like a sensory experience, sort of like misophonia, right? I don't know if you're familiar with that condition, but it's basically like just this like visceral, awful reaction to a specific sound. Like one of the common things that cause misophonia or misophonia in some people is like mouth sounds, like hearing people eat with their mouth open, that kind of thing. Anyway, so it's sort of like that. You're just like, oh, this is so awful, and it is just commanding your attention when a baby's crying. But I think that, while her limbic system is coming online, part of it is reward connections or reward associations. So, like, I do X and then Y happens, right? And so if she learns that when she just, pretend cries, obviously I'm not like, ignoring, if there's a good reason for her to be crying, I don't ignore that. But, if she like doesn't get to, whatever, just eat ketchup with her fingers, and she starts crying, I'm just like, no, sorry. This behavior generates a lack of attention. And it's pretty cool. Like, just over the course of yesterday, I watched it work, where like she would start crying and I would just sort of like turn my head, go on my phone or something like that, and then she would like almost immediately stop crying. So just a little tip for those of you out there who are who have young babies. How do you memorize things easily? Because I have a very, very forgetful brain. Alex Fawny, that is an awesome question. And in fact, I wish that it was like, and I'm sure like this, every college student that begins majoring in neuroscience, they immediately ask, this is the first question they ask, like, hey, how do I learn better? Because you're forcing me to learn all these things at this crazy clip. And what's the cheat code? Give me the cheat code, professor. And the answer was, honestly, I don't know. And here's how I do it. First of all, I don't have a remarkably good brain. Like, it's not like this, unusually, brilliant mind. My memory is pretty much run of the mill. For some things, I have a terrible memory. Like, I'm very bad at remembering people's faces. And so, yeah, I had to come up with some kind of, particularly when I went to graduate school, like, I had to memorize the names of like dozens of genes and how they interacted with one another. So here was my strategy. First of all, try and explicitly associate positive emotional valence to the information, right? So that's, probably sounds like gibberish, but basically it's like there is a reason you're trying to learn something, right? There is some reason. It could be as simple as, getting a good grade on a test, or it could be that you want to memorize a talk or whatever, just like, information that you have deemed necessary to learn for one reason or another. But the human brain, like, the primate brain, evolved. The reason we have emotion is a sort of behavioral rudder, right? So if you anticipate negative emotional experiences subsequent to any given activity, then you will avoid that activity, right? And conversely, if you anticipate good emotional experiences after a given behavior, you will approach those behaviors. And so the human, there is no such thing as a Vulcan brain where it's pure, like a lack of emotionality, right? So emotion is extraordinary, mood is very extraordinarily important to human behavior. And so as a result, if you're like sitting down trying to memorize this like sterile list of genes or whatever, be very explicit about the emotional reason, like the cosmic reason that you're trying to learn this. Are you trying to learn this just because you want to get a good grade? Why do you want to get a good grade? Do you want to, make sure that you have a great GPA, or you want to make sure that, you're prepared to, excel in a given profession? Why do you want to do that? Well, because, it'll make life better for you, theoretically, but also for me, like, part of it is that I want to make sure that I do the best that I can to provide circumstances for my daughter, for my children, that are optimal, so that they'll have the best possible opportunity to do the same for their kids. And so, all of a sudden, this list of, sterile genes becomes like this dire, important, critical thing that you have to learn. And, that'll sustain you for a little while. Another thing is, so much of why human intelligence is as remarkable as it is that we had to socially cooperate as, evolving primates. And so as a result, like a lot of cognition or trying to learn new information, devoid of any kind of anthropomorphic kind of component, that, trying to learn information that has zero human investment or zero human characteristics can be very, very difficult. But if instead of, again, just like running down this list of genes that you have to memorize, if you sort of devise a kind of play, a kind of like, imagine what it would look like in a movie where each of these genes has a certain characteristic, maybe they look like a certain character or whatever, right? Try and anthropomorphize it. That can, at least for me, that helps a lot. And the last thing is, this is much easier. I found that if I was exercising while trying to learn new information, that helped. So I would study on a treadmill, for example, or I would literally, in college, I managed the rock climbing wall. There's like this small rock climbing wall in our gym, and I mapped out all the courses. And so I would just like tape note cards to different parts of the wall, and so I would have to get to a certain point to get to the answer, and it probably just made it more fun for me. But I do suspect that there was like an additive benefit to doing that. And I see DC Hooper 91, Deanna Hooper is in here. She is a cosmologist, a scientist of the cosmos, and she studies dark matter, and she does this kind of thing. So check her out, DC Hooper 91. What are the effects of alcohol on the brain? Oh wait, was somebody trying to call in? Darn. I think somebody was trying to call in. Sorry about that. What are the effects of drinking alcohol on the brain? Well, they are. It's a diverse set of effects of alcohol in the brain. So first of all, relative to pretty much any other drug that people do for fun or voluntarily, we're talking nicotine, we're talking cocaine, we're talking cannabis, we're talking, heroin. Alcohol is what we call much more promiscuous from a pharmacological perspective, not because it makes you more promiscuous, although it can. And that's part of, and that results from its pharmacological effects. So when I say promiscuous, it interacts with a bunch of different components of the brain. Probably the most commonly cited pharmacological effect of alcohol is enhancing gabaergic transmission. So GABA is the brain's principal inhibitory or off neurotransmitter that makes it more difficult or makes it less likely that a neuron will fire an impulse, fire an action potential. And so, globally, alcohol suppresses neuronal activity. And there's a bunch of other things that it does. It interacts with, glutamate receptors, serotonin receptors, mu-opioid receptors. In fact, there was a study, and I always like to cite this because I think it's probably surprising to some people. It turns out that if you block mu-opioid receptors, those receptors to which morphine, heroin, oxycodone bind, if you block them, people will stop self-administering. People with alcohol addiction will stop self-administering alcohol. Pretty interesting, right? An opioid receptor seems to be critical to alcohol pharmacology. Okay, so all of this enhanced GABAergic activity, what happens is different parts of the brain become, they have less signal strength. They're emitting a sort of a lower volume of activity. And there's a bunch of different brain regions that are all involved, but let's just do two that I think are the most interesting and perhaps the most important. So the first is it inhibits a part of the brain called the rostral-medial tegmental nucleus, or RMTG, which is a fairly recently identified GABAergic pretty much component of a dopaminergic part of the brain called the ventral tegmental area. So what does that do? That makes, and it is sort of the brakes on the VTA, on those dopamine neurons. And so if you inhibit it, then all of a sudden those dopamine neurons are firing even more, right? And so that is probably the prime mechanism by which it is rewarding, why people even like alcohol. But then the other one that I think is most important from a behavioral perspective is that it inhibits frontal inhibitory output. And so, whenever you experience you could just sit in your chair and eat like jars and jars of cookie dough, right? That would be great. It tastes lovely, and it has a lot of calories. But yeah, it tastes so good. So why don't we do that? Well, there's certain parts of the brain that those urges sort of crop up, like, hey, why don't we just eat a bunch of cookie dough? Like, why are we not doing that? Then frontal regions, there's other regions too, but frontal regions will stand like, shut up, this is such a bad idea. Like, we don't want to develop diabetes, which I talked about on YouTube today, youtube.com/anthropoid. Anyways, so it inhibits those regions as well. And so that is part of the reason for why people who have had some alcohol can behave a little bit more recklessly, maybe a little bit more courageously, right? There's a reason that it's called liquid courage, is that it inhibits those anxiety-provoking, anxiogenic parts of the brain. So over the long term, we can talk about the long-term effects of alcohol if you'd like, but they're bad, all right, oh man, I'm behind. Okay, what can we do to regain brain cells? Well, testadura. There's really not much you can do. Like as a single human, can do on your own. There are parts of the brain that are generating new neurons as we speak right now. And a certain part of the hippocampus called the dentate gyrus. Not all of the neurons in that region, but some of the neurons of that region undergo neurogenesis. And then there are certain regions of the striatum that do the same. But by and large, you lose a neuron, it's gone forever. Now, what can we do to regenerate neurons? Well, in fact, I think last week, was it last week or 2 weeks ago, I talked about a clandestine operation that was performed, an experiment that was performed on a rather wealthy, independently wealthy physician who developed Parkinson's disease. And they infused 4 million neuroprogenitor cells into the part of the brain that causes Parkinson's disease. And there have been other studies apart from, this one, and the reason it was clandestine is that nobody knew it was happening. And I think most people in the field would have been like, I think it's a little early to be injecting stem cells into the brain. But there have been animal studies that show that when you do that, when you infuse, these neuroprogenitor cells, meaning like stem cells for neurons, essentially, these are cells that can become a bunch of different kinds of neurons. When you infuse it into a damaged region of a rodent brain, the neurons not only differentiate, we call it differentiate, they not only become neurons, they become the right neurons, dopamine neurons, and they form anatomical connections with all the regions that they would have to be able to effectively cure Parkinson's. So that is neuroprogenitor cells, stem cells, regenerative medicine in general. That is almost certainly the most likely means by which we will regenerate human neurons, central nervous system neurons. How does CBD affect the brain? Stay easy. That's the handle of the person. I'm not asking you to stay easy. So CBD, cannabidiol, is probably the second best understood cannabinoid molecule in cannabis. Very creative naming scheme we have going on with cannabis. But anyways, so just like tetrahydrocannabinol, which is, or delta-9 tetrahydrocannabinol, which is probably the active ingredient of cannabis with which you're most familiar. A lot of people who have no interest in science or neuroscience have any, they've probably been exposed to THC, or at least the word or the initialism. So CBD, so THC binds endogenous cannabinoid receptors that are quite densely expressed throughout the central nervous system and some in the peripheral nervous system. But the central nervous system includes the brain and the spinal cord. So THC is thought to be as a result, THC is thought to be responsible for the vast majority of cannabis's psychoactive, psychedelic, state of consciousness altering effects, whereas CBD binds CB2 receptors. So THC binding CB1 receptors, most widely expressed in the brain. CB2 receptors, cannabinoid 2 receptors, are more widely expressed in the peripheral nervous system, which is why CBD, cannabidiol, is not thought to really have very significant psychoactive effects because most of its effects are occurring, pharmacological effects are occurring outside of the brain. However, two things. So first of all, I have noticed like a lot of cannabis dispensaries, since cannabis is now legal to consume just for recreational purposes in a lot of states across the country, a lot of these dispensaries will sell like strains of cannabis that are thought to be, either by far have higher CBD or than THC or no THC at all. It is totally possible that such strains exist, but if I were a betting man, I would bet all my Bitcoin on that most dispensaries are not really performing any kind of analytical chemistry to determine whether or not there's any THC, in these CBD heavy strains. So that's the first. Could totally be real in some cases, but by and large, my suspicion is that it's not. But generally speaking, these are people who smoke or consume those strains, and they're like, they have a certain effects profile. And they're like, oh, that must be CBD, but it's pure speculation. Okay. The second is that one of the most common reasons that people use CBD is to treat anxiety. It is thought to be anxiolytic or anxiety reducing. And this has been shown in at least some people, it has been shown to exacerbate anxiety in some people. So, it's not going to be like consistent. Yeah. And pain, although same story, like it has been shown to reduce pain in some people, but also exacerbate pain in some people. And we can we can get into the details of some of those, or some of the explanations. I studied at the University of Pennsylvania. So if any molecule is reducing anxiety in a way that is, substantial enough to be like this large group of people are saying, hey, this reduces my anxiety, that's a psychoactive effect, right? One could make the argument that maybe it's a peripheral effect where it's reducing, the release of cortisol, glucocorticoids, or maybe adrenaline in the brain called epinephrine, maybe, and that would have this sort of indirect anxiolytic effect. But my bet is that, it crosses the blood-brain barrier and it binds some receptors that reduce anxiety. But that would make it, so that would make it like technically psychoactive, but relative to like THC, significantly lower psychoactive effects. So significantly lower effects on the brain. Liquid chromatography is easy to do, so they could quantify a percent of CBD and THC if they. Yeah, sure. Find me a dispensary that is doing that though. I mean, again, I'm sure they're out there. I'm totally sure they're out there. But I lived in California for a while, and I'm doubtful. I'm doubtful that they would do that. Do you remember at what age GABA turns inhibitory? BC. Because it starts as excitatory, if I recall. So yes, I think it is puberty. I think is like, and so it'll range in different people. We all begin puberty at different times, but so around the beginning of puberty, specifically within the hippocampus, I don't know if GABA receptors are excitatory everywhere. Yeah, so what Abyss is bringing up, it's this really cool phenomenon, and GABA receptors are not the only ones. In fact, cannabinoid receptors do this. But what happens is, as I was saying much earlier on, GABA receptors tend to be inhibitory. They're not always inhibitory. Like, you can find excitatory GABA receptors in the brains of adults, but generally speaking, they're inhibitory, meaning that they make it less likely that a neuron is going to fire an impulse or an action potential. However, earlier on in life, this is when we're adults, earlier on in life, particularly in the hippocampus, GABA receptors are actually excitatory. This is one of the reasons that it is thought that people can grow out of epilepsy. Epilepsy, of course, is a condition where people have recurrent seizures. And one of the things that can happen with a child who has epilepsy is that they can, as they get older, just sort of goes away. So that's one of the theories, that there is a lot more excitatory activity in the brain of young people than there is in adults. So there's that. Now, what's interesting is, what's additionally interesting is that cannabinoid receptors also undergo this age-related sort of swap in signaling. So again, cannabinoid receptors, either receptors that THC, CBD bind when somebody smokes or eats pot. These are the receptors that are being stimulated. It turns out specifically, it probably happens elsewhere in the brain as well, but in the amygdala, there is this reversal of the sort of signaling cascade when cannabinoid receptors are stimulated. And so it's sort of this like classic thing where somebody may have smoked weed when they were, younger, whatever, and they really liked it, but they stopped for, one reason or another. And then, they retire, this, particularly with the hippie generation, right? They retire and they try smoking pot again and they lose it. They freak out. they have severe anxiety. They can have a panic attack. And, generally speaking, it's probably because the pot that they were smoking when they were younger was like almost a different drug altogether compared to the cannabis that's generated today, which is, vastly more potent. So there's that. So they're probably in part overdosing, particularly if they eat it. But also it could just be the case that the effects of cannabis are different in the brains of adults. So it's kind of a cool thing. Neuralink. Are we going this direction? We can talk about Neuralink if you like. It's one of my favorite topics. Yeah, I mean, is it realistic? Yes. But I, like everything Elon Musk does, I'm skeptical of their timeline. I don't believe that they're going to have this in the brain of a human anytime super soon, or at least not have a whole brain computer interface. What I do think is that, and it's totally realistic, is that they could devise a tool, a Neuralink, a device that is significantly superior to what the sort of standards are, because we implant stimulating very, very simple brain-computer interfaces, right, to treat various things, Parkinson's disease. But so I bet I'm not skeptical that they could generate a better version of what exists today. In fact, that is their plan, to begin with that. So yeah, that's realistic. But the idea that they're gonna generate a whole brain-computer interface anytime too soon is a stretch. And in fact, I saw a story in Stat News where a lot of the original scientists that composed the board of Neuralink, the sort of founding board, have left. I think it's something like there were like 7 or 8 of them, and now there's like 3 of them or 4 of them, and it's just because they have some quotes in the Stat article, but like they were just saying this is completely unrealistic. Like the pace is completely unrealistic, and there's this tension between the engineers at Neuralink and the neuroscientists, which happened. I mean, that's natural, but evidently this was just prohibitively like over the top. And Elon Musk, being an engineer, usually sided with the engineers. So I can imagine that being frustrating. But so that's a problem. But hey, that's inevitable, and it's totally solvable. Okay, where did I say? We talked about that. Can brain cells regenerate? Yes, they can. There are certain parts of the brain where it's happening in you right now, depending on your age. There could be more or less, but by and large, they are the exception to the rule that central nervous system, meaning brain, spinal cord, neurons, do not regenerate. Can I still get high on Neuralink? Can you get high? Well, okay, like if you have a whole brain computer interface, could you get high? Like, would you need to take a drug? The answer is no. Like, so again, this is still science fiction, right? That we do not have a device like this that's available, not even close. Oh my gosh. Not even close. But let's just stipulate. we've made this device. That means that you could theoretically download stimulation protocol from the internet, port it into your Neuralink, and then your Neuralink and the this downloaded file, stimulation profile, could be labeled cocaine. And so, you don't have to take any cocaine, you don't have to snort it or inject it or smoke it. You could just download the stimulation pattern and then, trigger your Neuralink to induce it. And then you're feeling like you're on cocaine. It doesn't mean that it would be less dangerous than cocaine, right? But I don't know if you can hear her, but yeah, I mean, and that's just one example. And by the way, like, that would be like the first generation of whatever company comes out with, the drug stimulation paradigm. But why do that? Like why go with cocaine? Why not improve upon the effects of cocaine? Why not make it so that there is no crash, or that the anxiety and the paranoia and the psychosis that can result from high doses of cocaine? Why not just get rid of that? So yeah, just like an iPhone camera filter. Yeah. So yeah, you could get high on it. Hey Haley, it's been a while. How you doing? Would you suggest CBD to help cure anxiety? No, I wouldn't. Nervin, not because I have anything against cannabis or CBD. As I regularly say, I'm totally in favor of completely legalizing cannabis for recreational use. Doesn't even have to have medicinal value, although it does for some patients, for some people. But rather, it's because, first of all, there seems to be a pharmacogenetic, meaning like a distinction in how people with different genes, right? You have different genes than I have different genes than my brother has, although we have more similar genes than you and I have, right? And on we go, right? And like some of those differences emerge at the level of neurotransmitter receptors. And being these big honkin' receptors, CB receptors, cannabinoid receptors, there are a lot of opportunities for genetic variances, or single nucleotide polymorphisms, or just like slight distinctions between the way that your cannabinoid receptor reacts to being bound by cannabinoids relative to mine. And so as a result, the effects of CBD are not super consistent, right? So some people do react with lower anxiety, and that's great. Go ahead and use it. But some people react, it exacerbates their anxiety. And I suspect that is the reason, that there are these pharmacogenetic, individual differences in how people react to all drugs. And that includes CBD. Could it release DMT? So I assume you're referring to Neuralink releasing DMT. Well, once again, so first of all, probably not. But there's a bunch of reasons for why that's the case, and also why you wouldn't even need to do that in the first place if you wanted to have a DMT experience. So first of all, DMT, dimethyltryptamine, is one of the members of the family of psychedelic drugs. DMT is a modified DMT molecule is found in psychedelic mushrooms or magic mushrooms. DMT grows naturally in a lot of different plants. Most commonly, it is derived from a root, or actually the root of a vine called Mimosa hostilis, and it's extracted, and it can be consumed in a bunch of different ways. It can be consumed either by extracting and then crystallizing it and then smoking it, or in a combination of a bunch of different, essentially stew, tea. And so that's generally speaking how people get DMT. But anyways, and it's psychedelic. Its pharmacology is kind of similar to psilocybin in magic mushrooms, or LSD, or mescaline. It's probably most comparable to psilocybin. Psilocybin is just DMT with a phosphate group attached in the 4 position. So as a result, they have slightly different pharmacologies, but more similar than like DMT compared to nicotine. Okay. So your question presupposes that first DMT is produced in the brain. So, and I know that a lot of people now are of this belief that DMT is produced in the brain, and not only that, but it is the DMT that's produced in the brain that's responsible for dreams, near-death experiences, and just the sort of more exotic, components of the human experience. And so, first of all, there is no evidence that DMT ever reaches levels in the brain that are what you might consider like meaningful. Like it has been found at trace levels, meaning very, 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 very small concentrations in various parts of the brain, not just the pineal gland or pineal body, and never at higher levels than that ever. And so that is probably here's why I think that happens. This is speculation, but I bet I'm right. That dimethyltryptamine, the reason it has these pharmacological effects, these psychedelic effects, is that it's quite similar to serotonin, the molecule, in sort of the shape of the molecule. In fact, probably to the untrained eye, it might be difficult for somebody to distinguish between DMT and serotonin. And so as a result, neurotransmitters, serotonin, dopamine, norepinephrine, all of these molecules undergo a bunch, they're produced by a series of chemical reactions. And then they are degraded through a series of chemical reactions. And so as a result, serotonin is formed by manipulating an amino acid called tryptamine, and then it will encounter other enzymes. Enzymes are what enable these chemical reactions to occur. Enzymes are what enable us to create dopamine in our brain or create serotonin. And as a result, here's what I think happens. It just by chance, in some cases, not the most common mechanism, but in some cases it encounters an enzyme that modifies serotonin in such a way as to create a DMT molecule for just like a little bit of time, and then it's gone, gobbled up. And so by monoamine oxidase or some other enzyme. So that's what I think happens. And so as a result, it may play some signaling role, but it's almost certainly playing that role within neurons, regulating something like calcium levels or maybe binding signal 1 receptors or something. But it's not like there's ever a moment, or at least there's no evidence that there's ever a moment where the brain just opens the floodgates and outpours DMT. Certainly we don't need it to explain dreams, we don't need it to explain near-death experiences, and in fact we have great explanations for both of those things without any need for DMT. The second is, if you're using a Neuralink and you want to have and again, this is science fiction, we're not even close to creating something like a Neuralink, but you wanted to have a whole brain computer interface, you wanted to have this psychedelic experience you've heard so much about, then what you could do is go to a company that would be able to sell stimulation protocol, meaning like patterns of, stimulate this neuron at this frequency, stimulate that neuron at that frequency, and on, and it would be extraordinarily complicated, but you download this protocol, you hit initiate in your brain, and then you're immediately having psychedelic hallucinations, and then it can be gone. So yeah, I know that was kind of like a long sort of tirade, but I think Neuralink is a fun way to explore a bunch of different topics in the brain. Oh, hey, looks like Javier is calling. Wait, let me get this ready. All right, looks like we're connected, Javier. Javier, our friend from the United Kingdom, what's on your mind?
Hi, very simple question this time.
I doubt it.
Very simple. Yeah. No, well, maybe. Perfumer brought to my attention. Some people avoid ethanol in perfumes. And I was just thinking, ethanol obviously is known to inebriate people, but the perfumer told me other alcohols can do the same thing. So I'm aware of a chemical called terts, amyl alcohol, so terts, pentanol. But he gave me the impression that every alcohol does this, that they are gabaergic. Is that true? Is methanol gabaergic and isopropanol? Because I know the reason people use ethanol is because for serious toxicity, you need a humongous amount. I think it's like 400 ml to kill a human of pure ethanol. Whereas with methanol, it would be, I think, 5 ml, enough to kill a person. So it doesn't make sense to choose methanol over ethanol. I was just wondering if this is the case. What about propanol? You can have a lot more propanol. I think it's about 200 ml before there's the LD50. So I'm just wondering, is it possible to become inebriated by having a different chain alcohol?
Yeah, okay. Yeah, interesting topic. I love it. Some pretty, old school pharmacology here. So I honestly do not know for sure what the pharmacological profiles of, isopropyl alcohol or any of these other. So just so everybody's on the same page, an alcohol, ethanol is basically a hydrocarbon attached to an OH group, a hydroxyl group. And there's like a ton of different alcohols that one can make, different shapes of those chains, different lengths of those chains. That's generally what distinguishes different kinds of alcohol. Ethanol, I think, is just 2 carbons, but then, you can get, butanol and propanol and, just a bunch of different, just depends on how many carbons are attached to that OH group. And so I don't know for sure that they all have the same kind of GABAergic activity. It wouldn't surprise me. I do know that yes, I think it must be isopropyl does induce some of the similar rewarding components. When we say reward, the parts that feel good of alcohol, we know that because there are cases of people who, for one reason or another, don't have ethanol at their disposal. They're, generally speaking, they have a drinking problem, and they will consume like hand sanitizer or things like that, and it achieves their goal. But the reason that you don't want to consume methanol or isopropyl alcohol is that they may or may not have that pharmacological effect, but they will have a bunch of other pharmacological effects that are toxic. And so, just to give you an example, if somebody drinks methanol, if somebody drinks methanol, there will be some psychoactive effect, but it'll do things like it'll damage photoreceptors, or it'll damage it can make you go blind. And interestingly, and this would suggest that they do have overlapping largely overlapping pharmacological effects. Do you know what the treatment for methanol poisoning is, Javier?
It's actually taking other alcohols, if I remember correctly.
That's right, drinking ethanol. Yeah. So it's kind of like an interesting little thing. And the reason.
I don't know why that is, but yeah, I did read that.
Yes. Yeah. So the reason that is that for whatever reason, the ethanol molecule will have an ability to bind those receptors, those proteins, those things with which the methanol at the moment is interacting more tightly, and so it outcompetes. It bumps it out of the way, and stops the toxic biochemical events or pharmacological events. But so the short answer is, my bet is that since ethanol is such a simple molecule, that it is something about the shape, like if you have an OH group attached to a carbon, and it is capable of contorting in a certain way, that it interacts, any molecule that has that will be able to interact with a lot of the same receptor proteins. So yes, that would be my bet. But the reason that you don't want to consume any of those other alcohols is that they are toxic in ways that ethanol isn't. And by the way, ethanol is toxic. So I mean, it's not like it's a completely benign thing, and like the only problem is that you have a hangover. It is toxic to tissues, including neurons. But it just so happens that these other alcohols are more potently toxic. Yeah. Wait. So what brought this up for you?
I was just very, because some people, for religious reasons, they choose to avoid ethanol, and for example, in hand sanitizer or in perfume, they might say, well, I'll use isopropyl, and then a perfumer whom I spoke with told me, well, actually, other alcohols have the same issue.
I gotcha. Okay. Yeah. Well, that's true, but they're just toxic in ways that ethanol isn't. As Paracelsus, taught us, I think a Swedish or Swiss father of toxicology, the difference between a medication and a poison is the dose. And so, you're right that it is that these other alcohols are toxic at much lower doses in many of the ways that ethanol is too. And so that's why you wouldn't want to consume it. But you're probably also right that there are enough pharmacological similarities that at least for those moments before you went blind, you would feel some inebriation. By the way, that is super cool that you're friends with a perfumer. Like, I've always found that profession so cool.
Yeah, he's an independent perfumer, very successful actually in the UK.
That's awesome.
Yeah, thanks. There was a question about another alcohol that I was going to ask you. I don't remember now.
Well, you can feel free to call back in, or we can talk about it next week if you like.
Yeah, I think I'll do that. I don't want to just stay here thinking for 2 minutes. I want more days.
Right on. All right. Thanks for calling in, man.
Thank you. Bye.
So Javier from the United Kingdom. We became fast friends because he asked questions about free will and was clearly informed. He wasn't just sort of like asking, do we have it? He had, he clearly educated himself on some of the philosophical arguments for free will. I probably should have brought it back up because, he inspired me to go back and familiarize myself with, Patricia Churchland's, with Daniel Dennett's, with, Steven Pinker, with, all these people who have touched on this topic before. Anyways, maybe we can talk about it next week. All right. Hey, what's up? Oleg, good to see you, man, on YouTube. Eleonora. Hi, Ian. No broadcast on the Periscope. Oh, no, I do. Yeah. I can't see your broadcast. Oh, is that right? Huh. That's odd. I wonder why. Because clearly some people are. Thanks for letting me know about that, Eleonora. I'll bring that up. Okay, let's see. Doc, you've gotten old since how long? Oh, wait, I'm sorry. Hey, Doc, you've gotten old since I last watched one of your streams. How you been? Not bad. Yeah, you're telling me. Isopropanol also has a higher boiling point. There's also that. Yeah, so if somebody was boiling you, that would be good to know. Can we talk about cognitive neuroscience? Of course. It's, to be completely, transparent, I am a sort of circuits neuroscientist. I focused on, the interactions of neurons with other neurons. Specifically, cognitive neuroscience generally uses tools like fMRI, or other ones too, EEG sometimes, with humans. And it's just, it's just a different domain of neuroscience, a less cool domain of neuroscience. Although to everybody outside of neuroscience, cognitive neuroscience is the cooler. Okay. Doctors yelling shots. Honestly. Okay, so I went to graduate school, right, to get a PhD, and there are other graduate, degrees that one can pursue, like a law degree, a medical degree, business, MBA, business degrees. I guess you can get a PhD in business. Some people do that. I went to parties with, all of them. I mean, and we're talking about graduate student parties, not like chug type parties. Here's the order of how, just the volume of alcohol consumed by these different disciplines. It goes law students. They're crazy, particularly like first years. It's wild to watch. It's just like, God, it feels like you're in a frat or something like that. Of course, these are just the law students that I knew at UPenn. It goes law students, probably then business students. No, law students, medical students, business students, and then us. But if you ever have a choice to go out for dinner or for drinks with any of those people, choose a neuroscientist. They're going to be the weirdest, the most interesting, and they'll be able to, have interesting conversations. All right. I'm obviously mostly kidding. Only mostly. All right. Alcohol is silly, lol. It doesn't even improve your life experience. It literally debilitates you, lol. Yeah, well, I mean, you could make that argument about basically every drug, right? You can make that argument about amphetamine. Amphetamine over the short term is performance enhancing, theoretically, on very, very specific tasks. And but there's, you have to pay the piper at the end. There's a withdrawal period where you're actually, less cognitively acute or aware and effective than before. But the other thing is that, the reason that alcohol is almost a critical part of human evolution. Obviously not every society or culture consumes alcohol, but a lot do. And, it's clearly served this purpose, mostly social, that has been useful as humans, migrated out of the savannas of Africa. So it does have some utility. He said better not to know. Oh, I'm 53 and asked a neurologist if I should do anything about my increase in forgetting why I walked. Why I walked? He said better not to know. So I guess I'm not 100% sure what you mean by why I forgot to walk, but I think you probably mean you're concerned that you're forgetting more frequently. That is definitely something that you should bring up. If you're consistently forgetting things, okay, where did I put my keys? why did I enter this room? You know, like if you're forgetting like that with you know, we're all forgetful sometimes more times than others, right? But if this is consistent for you, definitely bring it up with your physician. Absolutely. Yes, Indigenous Australians have ridiculously low alcohol tolerance, not part of their culture. Indeed, yeah, and that's true in a lot of, you know, various parts of Asia, very commonly, I think, Southeast Asia. So common, in fact, that they, you know, for a while it was termed the Asian flush. Because they lack a certain enzyme, alcohol dehydrogenase, or that the enzyme that they have is, I can't remember if it's more effective or less effective, and it just basically causes like an immediate hangover. So yeah, that's true, but they're a small proportion of the broader, you know, 7+ billion of us. But you know what you would call that? You would call that protection against alcoholism. All right, walked into room. Yeah. I have a question. Please accept first. I conditionally accept you, just like some governors can conditionally veto legislation. I conditionally accept your question. All right, what is consciousness? That's a great. That sounds like a super simple question or topic. It's actually, it has bedeviled neuroscientists for a very, very long time. And depending on with which neuroscientist you're speaking, you can have different answers. For me, the way I think about it is consciousness is an emergent property of the brain, of a brain. And what this enables, so an emergent property meaning it is a thing that is the byproduct of a bunch of different brain regions, different, you know, combinations of brain regions, all interacting simultaneously or almost simultaneously at very, very similar times, or, you know, when activity is precipitated, that produce consciousness at different levels of complexity. And so, you know, for example, like one philosophical like phrase that is used to describe this is like a personhood, like a gradient of personhood. So as opposed to like only humans have consciousness or, you know, personhood, they're slightly different concepts. But you know, some people believe that like, you know, a dog doesn't have doesn't have consciousness. You know, it can behave, it can do various things, but it's not conscious like humans are. But then others argue, and I agree with this, that there is just a spectrum of complexity of consciousness or comprehensiveness of consciousness, that and the you know, point along that spectrum, and humans are at one end, and then the rest of the animal kingdom falls or you know, falls behind, and depending upon how similar your brain is to a human brain, you know, you can have a more or greater consciousness or lesser consciousness. So I read that crows may actually be self-aware. Yeah, I saw that there are a couple news articles. I forgot to read them though. Now I can see via Twitter. Huh, that's odd. That's irritating. Oh, it looks like somebody's calling in. Oh, it's Javier. What is your avatar, Javier? Is that food?
Yeah, I think everyone says that. It's actually cocoa butter.
Oh, nice. Okay. So what's up? Why'd you call back in?
So I remember I saw a like an infogram, I think they call it, not sure, picture article, and on it was the effects of ethanol on the body, and related to your field of study, there's a strong link I read between ethanol and dementia increases the risk of it. I read the number was 100%. So I was just wondering, what is that? Like, because I thought dementia and Alzheimer's have to do with these proteins that build up in the brain. And so what is ethanol doing with these proteins? I don't understand that.
Yeah, no, that's a great that's a great topic. So yes, there's absolutely a connection between ethanol, or, you know, alcohol consumption and cognitive decline and dementia. So dementia is a broad umbrella term. Alzheimer's disease is the most common cause of dementia, but there are other causes of dementia. And as you noted, Alzheimer's disease is a byproduct, or at least we think it's a byproduct. It is certainly marked by, or characterized by, the aggregation of these misfolded proteins or miscut proteins, amyloid beta and neurofibrillary tangles. And one of these accumulates within neurons, the other outside of neurons, and it just sort of gets in the way. It disrupts the ability of neurons to communicate with one another. And dementia is this sort of broad set of symptoms that basically all arise because different parts of the brain are just not communicating with one another as they normally would. It could be because these amyloid beta plaques and neurofibrillary tangles are aggregating in the hippocampus, and so as a result, the hippocampus is one of the most important brain regions. As a result, all of the things that the hippocampus participates in, mood, memory, spatial reasoning, all of these things become disrupted. So that's Alzheimer's. But you can have other forms of dementia where those types of aggregates, you can have Lewy body dementia. These types of aggregates tend to accumulate in different parts of the brain, and so you can have slightly different manifestations of dementia. Now, all of that. So those are like specific diagnoses for dementia. But just, dementia arises because there is this breakdown in the ability of different brain regions to communicate. So anything that causes that will cause some kind of dementia. And so, as, we were noting, alcohol or ethanol is toxic. Like it's kind of wild that ethanol is like essentially the only legal drug to consume for fun in a lot of countries where, because first of all, it does damage neurons, and so you're going to have a loss of neurons that is, irreparable. And other drugs don't do that. So yeah, it's just nuts. So like cannabis, for example, doesn't cause any kind of loss of neuronal, or it doesn't cause any kind of neuronal cell death, at least as far as we're aware. But there's plenty of studies that indicate that's the case. So as a result, alcohol will induce, the loss of some neurons, and that can generate, a form of dementia for sure. I'm not sure, like 100%. I'm not. That definitely doesn't sound right. But maybe it's a slightly different statistic. So yeah, but that's basically why. The thing that the amyloid beta plaques, neurofibrillary tangles, the various aggregates are causing in Alzheimer's disease, frontotemporal dementia, all these forms of dementia, what they're generating is a breakdown in neural communication. What long-term high-dose alcohol consumption can cause is the same thing. It's just not via the same mechanism.
Yeah, that makes sense. I guess this is more of an opinion thing. Like, knowing this information, do you find in yourself, and maybe if other people hear this information, do you not think that they would be a bit more cautious about, drinking?
Yeah, I mean, I think so, absolutely. I mean, so it's an interesting topic. So first of all, for a very long time, biomedical science, that includes scientists and physicians who are not scientists, but they would basically indicate that heavy alcohol consumption is bad for you, right? Like at no point did in modern neuroscience or modern biomedical science, at no point did we ever think that heavy alcohol consumption is good. But for the longest time, it was argued that moderate or mild alcohol consumption is probably almost certainly benign, but maybe even beneficial, right? So the relief of stress and cardiovascular effects of like red wine, these were things that were highlighted as you don't want to binge drink or you don't want to drink heavily, but this lower consumption is okay. Well, a study that came out of the UK, in fact, I think actually it was Cambridge, I can't remember, basically showed that even mild alcohol consumption has deleterious effects on health. So that has sort of changed things. But in medicine, physicians are sort of notoriously slow on the uptake. Like, they're not still in school. They're not necessarily, their job doesn't require them to continue learning new information. They should, but and many of them do. But so as a result, a lot of physicians will make recommendations that are based off of scientific findings from 10, 15, even longer years ago. So I think that's one of the problems that we've only recently really come to appreciate, like just how bad alcohol can be. But then also, the UK government, the United States government, and many other countries, alcohol is the only legal drug. And so humans, generally speaking, are going to seek to alter their states of consciousness, whether it's drugs, psychoactive drugs like alcohol or cannabis or whatever, meditation, extraordinary, athletic, feats of exercise and stuff like that. Humans, and it seems like a lot of animals, are driven to alter their states of consciousness. And when alcohol is the only legal one, the cost-benefit analysis that anybody's going to do is going to be based off of, hey, I really want this, and so there's, and also I think there is just this implicit assumption that if alcohol was so bad for you, then why would it be legal? After all, cannabis isn't legal, and I keep hearing that cannabis is pretty benign, and maybe a given person tried cannabis when they were they shouldn't in high school, but maybe they did, in their younger 20s or something, and they said, this didn't turn me into a brain-eating zombie. And so, if cannabis is illegal, then, and alcohol is legal. So I think that there's just a cognitive dissonance in our drug laws that is very, very damaging and totally counterproductive. But ultimately, yes, I agree with you that our drug laws should be based off of biomedical science and not just these cultural prohibitionist drives that seem to, underlie our drug laws.
Yeah, I've heard this argument before, and it's very interesting how the science, it seems to keep changing its stance on alcohol consumption for moderate and light use. So I do recall when that came out in the news in the UK that even a small amount can be deleterious to health. But prior to that, people were saying that it was, it's fine having a glass now and then, it's okay. I was a bit suspicious about people making that comment because, of course, wine has, sugars in it and flavonoids, beneficial, chemicals. I suspected maybe the ethanol is in no way beneficial to the human system. It's just the other components that are actually what are being seen as having a positive effect on human health. I don't know if that's true.
Right. So by and large, I mean, I think that is a totally fair, that's a totally defensible perspective on this. So the issue with like red wine being healthy, a lot of that came from, well, first of all, some just statistics showing that there appeared to be lower incidences of various cardiovascular diseases occurring in people who drink wine, red wine specifically, every now and then. There is this thing called like the French paradox, I think is what it's called, where French people tend to eat a lot of cheese because some of the best cheese in the world comes from France, and they tend to drink some wine, they eat some bread. It's like the French diet is not the like hallmark of like what most physicians would recommend as a healthy diet, right? And I don't blame them. Some of the best food one can consume. But like, epidemiology and like disease statistics in France suggest that they're healthier than like Americans or people in the UK or a lot of different countries that have healthier, theoretically healthier diets. And so there was that was one of the early inspirations for, hey, maybe, and the French diet calls for red wine. Similarly, the Mediterranean diet, which is the sort of diet that most physicians would recommend, most scientists would recommend, or like a slightly augmented Mediterranean diet, that also calls for red wine every now and then. Think Italian food, or I don't know, Cypriot food. I'm not quite sure. Greek food, and so that was part of it. Another thing was actually I mentioned his name earlier. Strangely, came out of the lab of a guy named David Sinclair. He's at Harvard. He's a geneticist who basically was like one of the first people to identify resveratrol as this antioxidant molecule with more biochemistry, more pharmacology than just being an antioxidant. It was shown to have all these effects on longevity in a bunch of animal models, and resveratrol is notably found. It's found in like the skin of a lot of different sort of berries and stuff like that, but very classically it's found in the skins of grapes that are used to make red wine. And so the thought was that, hey, maybe that's the thing, that people are drinking this red wine every now and then, and maybe it's the resveratrol. The problem with all of that is that the amount of resveratrol that one would need to take to have like a substantive biological effect is so much higher than you would find in one glass of red wine, right? You're talking about like fractions of what would be in like a tablet of resveratrol. Second of all, we have not proven that resveratrol. I don't think that there's a consensus that resveratrol is like the wonder supplement, the wonder drug that some people seem to think. Okay, so there's that. And you're right. There's like flavanols and flavonoids and like pigment molecules that have interesting biological effects in something like red wine. But yeah, I mean, by and large, I think it's not particularly controversial to suggest that yes, there is no like in terms of the balance of potential health qualities of ethanol. Again, reducing stress. Reducing the release of stress hormones, or for some people, it is anxiolytic. It does reduce anxiety, and so for some people, it might be the only way that they could have met their wife or their husband is that they were slightly inebriated, lower anxiety, lower inhibitions, and so they struck up this conversation with the person they ended up starting a family with, right? So there's like all of those things that are reasonable to argue in defense of ethanol, balanced against the fact that there really have not been any biological, specific mechanistic arguments in defense of ethanol. And so, and the other benefits of other, beverages like wine and stuff like that just are not significant enough to justify recommending ethanol as a, like a supplement, like a nutritional supplement. So yeah.
It's very interesting. For the like pharmacological effects, the neurological effects of ethanol, is it the molecule itself, ethanol, that is damaging to the neurons, or is it the effects that the ethanol has, where there's like an excess of a release of a certain chemical? Is it both that are dangerous, or is it just one?
So ethanol can be dangerous in several ways, right? So first of all, the most proximal cause of a fatality derived from ethanol is overdose, particularly when combined with other drugs like benzodiazepines. Or notoriously, benzodiazepines are opioids. All of these drugs work, sort of synergistically to suppress respiration, and so people asphyxiate. Basically, they stop breathing, and this is after they've already passed out. That's how people die from opioid overdoses. But you only need lower doses of these drugs can induce that effect when combined with ethanol. So there's that. Of course, there's the organ failure, liver toxicity, kidney toxicity, that can happen just because they've been exposed to this molecule for so long that it begins to kill hepatocytes, and stuff like that. So that's like those are like the proximal dangers of ethanol. I don't think that there's any evidence that ethanol causes excitotoxicity, which is what you're talking about, where a neuron dies because it has been driven to fire impulses at a frequency that it never naturally would. So this is sort of very infamously associated with drugs like amphetamine, methamphetamine, cocaine, where dopamine neurons, the molecule dopamine itself is really quite reactive, and so as a result, it's very closely sort of regulated. But so if you just have this, so there's not only that, but like if you're driving a neuron to fire it at frequencies that high, superhuman frequencies from like amphetamine and cocaine and stuff like that, for a long enough period of time, that will kill those neurons. However, I don't think there's any evidence that ethanol does that. Rather, it is just a direct effect of ethanol on, various organelle systems within cells, disrupting cellular membranes, and those types of things, like direct biological effects of ethanol. Yeah. So I think that's generally the cause. Yeah.
So it's really the molecule itself rather than the effects of like what it causes.
Yeah, pretty much. But like another thing that it does, alcohol has all these, these very disparate effects. Like one of them, for example, is that it will, for somebody who's drinking, chronically and pretty high doses regularly, it can modify the way, your gastrointestinal system absorbs nutrients. And so, that can be that's sort of a negligible problem for, once every now and then. But, if it's like every day, and you're at doses sufficient to have a meaningful reduction of the absorption of various nutrients, then it basically means that like you're malnourished. You're sort of artificially malnourished. You could have the best diet in the world, but you're just not absorbing some of those vitamins and minerals, and as a result, that can have rather indirect but definite deleterious consequences. But yes, generally speaking, it's the direct effect.
Yeah, there's also the other on the infographic, I think, that it can speed up aging. So then this relates to I think it's called the telomeres.
Yes.
I think they shorten. I heard that being labeled under the like it being a theory of aging, that the telomeres shorten. It's being correlated, but I don't know if that's the reason for us aging. And I think I read a paper where ethanol, it has a chemical effect with DNA, and it binds to it in a certain way that is very bad for the cell. I was just wondering what you think about this theory of aging, about the telomeres shortening, and why ethanol would have an effect on aging. I mean,
Sure. Yeah. I mean, it's that's great. So, God, I feel like today is David Sinclair Day. So you're right. So first of all, so everybody's on the same page. Well, what are telomeres? So probably most of us are familiar with the term chromosome, right? We have a bunch of them. They're like these little squiggly X-like shapes, and they are the structures into which all of our genomes are stored, or in which they're stored, by which. Basically, your genome is just this long stretch of nucleotides, A, T, C's, and G's, and it's they're coiled and then coiled again and then coiled again, sort of like if you were ever a kid and you took like a long string or something like that and you just started twisting it like this, it'll start folding in on itself first, and then you keep going, then it'll fold in, the twists will fold in on themselves, and on it goes, right? It forms this like bigger, more rounder structure. And so those are what chromosomes are. And literally they are composed of things that hold our genetic material and our genetic material, 3D structures. Okay, so there is a problem with the way that the various mechanisms that express our genes evolved. So, we're fortunate to be living a fair bit longer than we have in the past. And so this was never a problem. There was never an effect of natural selection on this deficit in the way that genes are expressed. And so this is the deficit. Every time a sort of string of DNA is replicated or, copied, just because of the way those enzymes, those polymerases, operate, you lose a couple nucleotides at the end every cycle, right? And so whenever your cells are dividing, you're creating new skin cells and so on, those chromosomes are losing a little bit of genetic material every single time, and that's a problem. It wouldn't have been a problem because generally speaking, most humans only really reproduce, let's say between late adolescence and, later and later these days, but, 40s, 45, that's sort of the window. And, until you're 45, this is not a problem. I mean, it can be a problem, it can cause cancer and stuff like that, but, broadly speaking, population level, not a problem. But now that we're living into our 70s, some of us fortunate to live longer than that, this can become a problem. So what has evolved to sort of prevent that process of losing that genetic material during every replication, the reason we can last until until the end of our reproductive age is that we have these stretches of protective genetic material. So they're sort of like helmets, or it's just sort of slack that you don't really care. It's not expressing anything. It's not responsible for any genetic expression, any proteins or anything like that. It is literally just elbow room. It's just like genetic material you can lose and it won't have any kind of horrible consequences. But once those telomeres are gone, then important genes will begin to be affected by that loss of genetic material, and so the reason that let's just say telomere degradation is a theory of aging, is that, telomeres will over time become shorter and shorter and shorter until they're essentially gone. And then that's why a lot the theory goes, that's why a lot of the age-related pathologies manifest, because you have this disruption in genetic activity of the important genes, but it just takes a while for us to get there. That's one theory. And as a result, there are labs that are interested in, there's an enzyme called telomerase that will regenerate those telomeres. And so there's evidence in some animal models that it does indeed extend their lifespan. And so that is one of the, endeavors within the field of longevity research to see if that'll work for humans. Let's see what else. Oh yeah, and then you're right. Yes, alcohol can influence genetic activity in general, and a sort of broader theory of aging is that we have all of these systems in our body that are active such that they catch errors. So let's step back. So all of those little machines I was talking about that replicate our genome, they are remarkably accurate. They almost never make a mistake. And when I say mistake, I mean associating a T with an A, right? A C with a G. But despite them being remarkably accurate, they're not perfect, and they do make mistakes every now and then. And actually, different species equivalents of these enzymes have different rates of error. It's kind of interesting. But and so we have all these chaperone systems, heat shock proteins, all these small enzymes and things like that can recognize aberrant protein behavior, aberrant that's the main one. And so if you have a protein, so let's see what can happen if one of these polymerases, one of these enzymes makes an error, and you get a T instead of a C or something like that, right? Oftentimes, it won't really matter. It's just this tiny, tiny little minuscule part of a protein that's kind of inconsequential. But every now and then, it can be a very consequential error that, makes a protein. A protein is just this long string of amino acids rather than nucleotides that then has a 3D conformation. It folds in on itself to make a certain shape, and that shape is very critical for it to be able to do what it needs to do. Speaking within the brain, like a dopamine receptor that is able to bind dopamine, right? And not only that, when it binds dopamine, it does the right thing. It lets ions flow or whatever, whatever the receptor does. And so if you have one of these errors, if you have one of these incorrect amino acids inserted into the protein, and the protein can't fold properly, then that protein is going to misbehave. It's not going to be able to accomplish, what it's evolved to do. And so we have all these error detecting, systems. Chaperones would be the most relevant for this conversation. And they do a remarkable job capturing and getting rid of all these errors in protein expression. But as the rate of those errors begins to increase over time, those systems can become overwhelmed. And then as a result, that's how you get, in fact, that's how you get something like amyloid beta plaques, neurofibrillary tangles, right? There are systems that recognize, misfolded or miscut amyloid beta or whatever, things like that. The ubiquitin proteasome system. All of these systems have a certain tolerance, certain capacity for correcting errors, but at a certain point, that capacity is surpassed, and that's when you get, carcinogenic cells or, things like that, misfolded protein aggregates, stuff like that. So that is like a broader theory of aging. Telomerase or telomeres are just one component of that story, but theoretically a rather important component of that story. And yes, alcohol does interfere with a lot of those processes, such that it can be carcinogenic, alcohol. But this is how a lot of things are carcinogenic, like the combustion byproducts in tobacco and stuff like that. Anyways, yeah, you took me on this. I took you. We went together on this long journey after that conversation. By the way, this reminds me. I learned something crazy the other day, and probably you all already know this. But do you know about the wood frog? the wood frog can freeze itself. Well, it can allow itself to be frozen, basically totally suspended animation, right? Like we're talking like 5 degrees, like super cold and literally frozen. And then, after the winter, after they begin to thaw, they just pop back in their normal froggy selves. And it's so crazy. And the reason being that they are capable of producing this sort of antifreeze molecule that prevents the types of damage that would occur when most every single biological system would freeze. Isn't that cool? It's so cool.
Yeah. I didn't know it was that they produced this anti-freezing molecule, but I was aware that the animal could do this. Yeah.
That's cool, man. Yeah. All right. Well, thanks for calling in. Always a pleasure to talk.
Could I just finish with one question?
Sure.
I've read something that lobsters are immortal. Is that totally false?
No. I mean, so there are some animals that, I mean, if not immortal, if not like completely, resistant to the effects of aging, then they live a very, very long time. Like there are whales, what is it called? It's called like the bow front whale that can live like hundreds and hundreds of years. Obviously tortoises and some turtles are, notoriously live for many hundreds of years. Corals, for example, they can live for thousands of years. Some trees, obviously, can live for hundreds and hundreds of years. So my understanding is that, yes, lobsters, the thing that limits their ability to continue surviving is not, the replication errors that we were talking about for humans. Rather, it's just they continue growing in size. And at a certain point, their like biological system, their the equivalent of their muscles, just cannot move like how big they become and heavy they become. So it's sort of like what would happen if you had a let's see like a a bird that was 700 feet tall, right? Or 1,000 feet tall. What would why couldn't that exist? Well, because bird bones are so brittle, they're like essentially hollow, and so they wouldn't be able to sustain all of the other stuff that goes into a bird body, and it just wouldn't be able to fly. It would probably just collapse, because those bones would just they're only able to withstand a sort of proportional mass. And so my understanding is that for lobsters, if they reach a certain point, they just can't move and they die. They are like an interesting model that definitely longevity scientists study.
Very interesting. Thanks.
Yeah, thanks for calling in, man. Always awesome to talk. That's Javier in the UK. I believe in London. That's why mammals can't do cryogenics. Water molecules form shards of ice, rupturing cells. Yeah, sort of. Yeah, I mean, there's that. But then also 1,000 points to, Euromeister's in here. 1,000 points to Gryffindor for anybody who can tell me what happens. What is it that makes water so weird when it freezes? I'm sure somebody in here definitely knows. But what happens is that it expands. It doesn't shrink. And so as a result, you have all these water molecules in things like neurons and, in blood cells and stuff like that. And so those cells just... And that's very frustrating for somebody in my position who, slices brain tissue, or sliced brain tissue, preserved it, froze it, and everything. Is that, one of the things you must do to avoid destroying your sample, your slices of brain tissue, is treat it in such a way that you try and remove as much of the water as you can, while still preserving its anatomical, conformation. So everything is where it's supposed to be, and neurons don't look all crazy. And I've mentored maybe 15, younger students who are learning histology, and always they come to me once they've learned the process of cryosectioning and stuff like that. They come to me and they're like, "Hey, my slices have all these holes in them," and it's like, that was the water. You didn't do your perfusion right. Super frustrating. Okay. So, great to catch you live. Oh, Lady Cosmic Rose. Yes, thank you. Great to be here. Speaking of alcohol, have you discovered any good beers lately? I totally meant to actually have a beer today on this. I have this beer called Unicorn Farts. I don't know if it's good. It's supposed to be quite good. But it has like edible mica in it. So it has these like little sparkles that evidently are not glitter in it. Yeah. Oh, so I now, one of the great things about living in Philadelphia is that we get all these awesome beers from the northeast. So I have a fridge full of Heady Topper right now. And I tried one called The Wrench, which is great. Oh, here's a great one. The Substance by the Birch Boy Brewery or something like that. Like the Birch Brothers Brewery. It's something like that. I can't remember. Bowery Brothers. And it's like clearly very like scientifically inspired. They have like the chemical structures of the main molecules that give hops and IPAs their like notorious flavor, but also their brewery, the initialism would be BBB or Blood Brain Berry. And that one's quite good. All right, I think we saw Abyss. Oh wow, okay. So Euromaestro must have called earlier because I just saw Abyss call in. So why don't we start? I hope you'll forgive me, Abyss, but why don't we start with Euro? So hey, Euro, what's up, man? What's on your mind?
Hey, how are you?
I'm hanging in there. How are you?
I'm doing well. So I joined late, so I apologize if you already answered my question, which you might have. But since you were talking about anti-aging research, I was wondering if you had maybe touched upon this research and studies that seem to show that caloric restrictive diets can significantly increase lifespan.
I love that you brought this up for a couple reasons. First of all, yes. So caloric restriction. Intermittent fasting, basically just limiting the amount of food that one consumes, either every other day or specific days of the week, like just fasting for the whole day, or something like intermittent fasting, where you only eat during a window of time in the day. So you don't eat 3 square meals a day. You only eat after 8:00 p.m. You gorge basically for dinner. And there are various versions of caloric restriction and this type of thing. But yes, there's a lot of evidence that suggests that first of all, like longevity, it extends longevity, it extends lifespan and health span. There are there's evidence that it has like cardiovascular benefits, cognitive benefits. It is a very, very hot topic right now. So there's that. And thank you very much for the super hearts. Appreciate it. But there was a paper that came out, I think, last week that purports to have done this study where they put like half of their study participants, can't remember how many, but it was an appreciable amount, on an intermittent fasting schedule. I think 16 and 8 I think was there. I can't remember. But anyways, whatever. It was intermittent fasting, and it was actually conducted by a physician scientist who himself follows an intermittent fasting eating schedule. So the short story is that their findings were that there were no significant benefits in weight loss or in muscle mass, and in fact what they detected was, in terms of the weight loss that did happen, most of it actually came from lean muscle mass rather than adipose fat. So basically just stored fat. One of the arguments that people make to advocate for intermittent fasting, caloric restriction, is that obviously it helps you to lose weight because you're just eating less if you're just doing caloric restriction, or it changes your metabolism in such a way as to promote fat loss with intermittent fasting. But one of the sort of main things that advocates will say is that it helps you to preserve lean muscle mass rather than getting rid of it while you lose weight. And so there's all of that. I myself have been doing intermittent fasting for a very long time at this point. I prefer it than just eating. I'm more sort of cognitively sharp, and it's not. This is anecdotal from my perspective, but there are studies that suggest that this is the case. Okay, so there's that. Now, what we can talk about is also why the sort of broad theory for why something like this would be biologically beneficial. And so, the short story is essentially, So first of all, humans may or may not have evolved to eat basically all day, right? Humans, they're my daughter's yelping. They spend part of the day gathering food or hunting or some combination of that, and then they go home and eat it. That may or may not be true. Maybe an anthropologist has a better insight there. But what it does is when a person is in a state of fasting and there's no dietary calories available to power a variety of different mechanisms, a bunch of survival pathways are activated. So this is also the argument for like exposing your body to like pretty radical changes in temperature, like using a sauna or going into an cryogenic whatever cryotherapy. Is that that activates a bunch of heat shock proteins, a bunch of pathways that do the very thing that I was talking to Javier earlier about, right? They detect aberrant cellular activity. It induces a process called autophagy, where a cell that's already that's already aberrant but still hasn't been terminated or gotten rid of. Being in this state, having all these pathways activated that are associated with survival, will increase the likelihood that those aberrant cells undergo autophagy and you get rid of them or you recycle them. Another thing that set of pathways that are activated is DNA repair. So if there's an error in any given genetic sequence, there are enzymes that can recognize those errors and either correct them or destroy that thing and make a new one. So that's the broad theory. I didn't like this most recent study that suggested that there was no benefit and perhaps even deleterious effects. And the reason is that, as far as I can tell, I mean, I very briefly looked at it. They literally the only intervention was some of them did intermittent fasting, and some of them had the standard 3 square meals type of schedule with snacking in between, basically eating the whole time. But there was no recommendation for any exercise, which obviously not everybody exercises, but if somebody's interested in intermittent fasting, then it probably wouldn't be too tough to convince them to also engage in cardiovascular exercise at a minimum, if not resistance as well. And I think that is like the critical component that is what really differentiates the metabolic, cognitive, blah 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 effects of intermittent fasting, and from it is the stress, the environmental stress of exercise that causes it, that distinguishes it. So okay, that I knew I was already going to go on this long soapbox tirade when you brought this up, just because I've been thinking about it for like the past 2 or 3 days. But anyways, yeah, that's caloric restriction.
Well, yeah, that was quite interesting. The particular study that I'm thinking of, that I had been read about a long time ago, was I don't think, as I recall it, really addressed the intermittent fasting part, but at least in lab animals, when they had these caloric restrictions, it showed an increase of like 33% in lifespan.
Yep.
So it's like significant increases, in like mice and rats and these types of things. So, anyways, I thought that was quite interesting.
Yeah, I mean, it is widely appreciated as the most consistent thing that can happen to an organism that will consistently extend lifespan. And you're right, it's been shown in a bunch of different animal models. It's been shown in worms and nematodes and mice and rats, in humans. And there's a bunch of other like interesting little kind of stories, like the effects of it's not like the notion of caloric restriction or fasting is like a completely exotic, out of nowhere idea, right? A lot of cultures include a period of fasting as part of their sort of cultural tradition or religious tradition, right? Like Ramadan, for example, or these types of things where they're basically intermittent fasting, and studies have been done on what the biological effects are of these types of diets, and essentially the evidence usually tends to fall on one side of this, which is it's beneficial, and so that's why I do it. I don't think it's appropriate for everything. Like, certainly if you're sick, you can make arguments both ways, but I tend to increase how much I eat if I'm sick. And I've been very, very fortunate. We all have that. We haven't gotten sick, particularly these days. And, you know, I would never have my child intermittent fast or you know, restrict her caloric intake for developmental reasons. So, you know, it is a sort of like, it's context dependent. But yeah, I find it fascinating and convincing. And so, yeah, that's where I'm at.
Well, anecdotally, I mean, I've been trying it myself for 4 or 5 months now, and so I've noticed a lot of beneficial things. I haven't really noticed anything harmful that I can tell. So focus, has increased, and certainly, you know, other types of like physiological benefits. So I've been pretty happy with it. The other thing is, you touched on briefly about these immortal species. So I have read that there's quite a few candidates for this, and some of them do it by different methods, which seemed quite interesting. So there's one that's nicknamed the immortal jellyfish. So it's a particular species of jellyfish. You've probably heard about it. But what it does is quite interesting. Is when it gets injured or it's sick, it ages backwards. So its cells actually go back to an earlier state that can then develop back into adulthood.
Yeah, I mean, there's, you know, species that do this where, you know, they basically you know, metamorphose they you know, they have their sort of jellyfishy type, you know, like floating type of situation, and then eventually they'll just form a colony of themselves, genetically identical. I mean, you know, to the outside observer it would look like it's just having babies and it's just planting eggs on the ground, but it's actually they're all genetically identical. And then boop, you know, they re-enter the, I forgot what they call it, but
like regenerative flatworms that you know, people are probably familiar
or like aspen trees, you know, like when you're looking at an aspen forest, it is like one tree, which is so crazy, very, very cool. But yeah, like in my mind, you know, like I'm all
There's
for.
a lot of odd things in the animal world.
Yeah, you know, I find longevity research really interesting. I actually literally on YouTube I talked about whether or not metformin might have some benefits on for, you know, cognition for people with type 2 diabetes. I did a podcast episode specifically on longevity. That's, you know, we touched on caloric restriction. We touched on, you know, nicotinamide riboside, mononucleotide, pterostilbene, resveratrol, all of these types of things that that are being, you know, evaluated for potential effects on longevity. But for me, and I think that's all valid, and it should continue happening. And I think it probably deserves more investment than it currently gets. But for me, I suspect that, you know, human immortality is going to come in the form of technological integration. You know, either integration or, you know, being able to regenerate you know, your consciousness in a computer. Now, it wouldn't feel like, you know, like you just your. Well, actually, we don't know this for sure, but my perspective is that it wouldn't feel like this contiguous experience. Rather, it would be like you now have a clone, and it just so happens that clone lives in a computer.
Unless they have some way of transferring the memories.
Yeah, well, but even still, like 100%, I agree. Like, you know, we're talking science fiction here, but like, you know, imagine I developed this platform that could, that could recapitulate not only, you know, every single neuron that you have, but where they are in the brain and to what other neurons they're you know, anatomically connected, and all of the functional properties of those neurons. Like, it's a perfect digital electronic representation of not only the human brain, but of your specific human brain. Therefore, all of the changes that you've experienced, all the synapses you've created, you've gotten rid of over the years due to your life experiences, they would be recapitulated in this representation of you. However, that doesn't mean that that your current personal subjective experience would that it would stop here and then begin on the computer. Rather, you are now split into two people, right? At the moment before it was downloaded, there was only one Euromaestro. But then, the moment that there is a digital representation, you've now split into two different people that could have very, very similar, very.
I totally agree with you.
Yeah.
But the second one, if he has my memories and experience, he he perceives, he has the illusion of continuity.
Yep, 100%. Yeah.
So he perceives it as a continuation of existence, and then depending on whatever happens to the first one, he has his own continuation, which is separate.
That's right.
So
Yep.
it's quite interesting. So I'm going to put you on the spot. What do you think the probability is over the next, let's say, 20 years, that not so much of the immortality, but that there is a major breakthrough in lifespan, extending human lifespan significantly, some sort of major breakthrough?
Did you say in the next 20 years?
Yeah, I'm gonna give 20 years because otherwise.
Well, if you were talking to David Sinclair, he would be, obligated to tell you, oh, it won't take that long, because they need funding, right?
Right.
But
Be a little bit more realistic.
Yeah. I mean, what we're talking about here is, this is a fundamentally interdisciplinary approach to understanding human biology, right? You need to understand nutrition, environmental, exposure to various, chemicals and sources of interaction with your biology, ultraviolet radiation, exposure to pollutants, exposure to, industrial chemicals used to produce our food. Like, all of these things are all critical, and they all arise or are optimally confronted by different disciplines. Not only that, but, to truly come up with a mechanism of some kind that will effectively extend lifespan, I think we will have to have a more completed genetics and neuroscience, and to better understand how we can competently intervene to correct certain aberrant activity, carcinogenic activity, blah blah blah. And we're just not there yet, particularly in neurogenetics. But do I think that I do believe that we have the opportunity to extend the average human lifespan pretty substantially. The problem, however, is that at the same time as we're learning more about things like caloric restriction, about various nutritional benefits, like we will within the next 20 years for sure, you will be able to have your whole genome sequenced, understand certain risk factors, compensate accordingly. You're at risk for developing lung cancer, you're at super high risk of developing lung cancer if you smoke. Well, then you don't smoke, and all of a sudden that limitation on your longevity is obviated, right? And so not only that, then you will have a signature of your microbiome, of your optimal nutritional levels, and what the perfect diet for you might be. We'll probably get to something much more like that, supplementing with probiotics that are actually effective and not just sort of speculated to be effective. All of these types of things will be available to you, so you'll have the opportunity to live much longer. However, at the same time, I think that there are other industries, other human endeavors that almost certainly impose a limitation on longevity. What I'm talking about are, the production of various goods and services, I guess, that generate a ton of pollution. And, but then also, that use chemicals that have a very significant toxic effect, or even a trivial toxic effect that over a long enough period of time causes somebody to die or develop cancer, etc. And so, like we learned about, I'll just give you one specific example that might encapsulate what I'm talking about. And when I talk about pollutants, I'm not even talking about climate change and that kind of stuff. I'm talking about we know that there's greater air pollution today, PM 2.5, very, very small air pollutants today than there was 100 years ago. A lot more, way, way more. And one of the most consistent let's say risk factors or predisposing factors for a child developing autism is exposure to high levels of air pollutants. This has been shown in Shanghai. This has been shown in, I believe, Mexico, in, I think, Los Angeles. It's pretty consistent. And so, while we'll be making all this progress over here, it's like if you're trying to clean a room, right? And you're making progress in one corner, and maybe let's just say you have a 1.5-year-old baby daughter, and you can't see her, but she's drawing on the wall with a crayon, and so you're making progress over here, but are you serious? This is even worse than it was before. So I think that is really the equation that we're currently navigating. And frankly, I don't think that the longevity efforts are winning. Also pharmaceutical, limitations. Like, for example, in the United States, life expectancy has decreased for like the past 2 years for a variety of reasons, but that is unacceptable. And there's no good reason for that other than it's not like we are worse at understanding biomedical science. No, it's these other things that are causing that problem. And so, I know this is like a very soapboxy, but that's how I see it. And so I don't know. I think there will be breakthroughs. I don't know if they'll be realized.
All right. So would you say that you suspect the net effect of those two factors that you talked about would mean that 20 years from now, lifespan would not necessarily increase significantly?
Okay, here's what I really think. Okay, there will be a contingent in various societies that has access to resources that are sufficient that they can insulate themselves from all of these issues, or many, many, many of them. They can live in pristine areas. They don't have to expose themselves to polluted air or polluted water. Their water is pristine. In other words, they can afford this. Whereas there will be another contingent of people who just that is not even a concern for them. Their concern is how I'm going to eat tomorrow, how I'm going to feed my baby. That's sort of where I see this going. It's sort of nightmarish, but I just don't see any evidence that it's not happening. So, yeah.
Europeans versus Americans, as I understand what you're saying.
Well, some Americans. No. Some Americans. Like, right, I would be willing to bet you $1 million that Jeff Bezos' water is like the cleanest in the world. Yeah.
But we'll probably see some effects too on the societal levels, like averages.
One would hope.
Different.
I certainly hope. Yeah.
All right. Well, thanks for that little age chat.
Yeah, it was fun. Good to talk to you, man. Thanks for calling in.
Yeah, bye-bye.
All right. I hope. Oh, Abyss, thank you so much, man, for hanging on. So we're talking to Abyss, the infamous Abyss. And let's see. That would be hilarious. Oh, no. Okay, we're connected. So what's on your mind, man? Hey, sorry for taking so long. But what's on your mind?
No, it's fine. So I guess it's kind of like tied to the paper that you talked about yesterday about stem cells, and it got me thinking like why progenitor cells went on iPSCs.
Yeah.
So did they explain anything on the paper? Because it seems like sort of like the common way to go.
Yeah, I mean, so I don't know. I don't think they did explain it in the paper. Frankly, I cannot remember though. I think generally speaking, the reason that one would do that is that, they know. So just so everybody's on the same page, Abyss is referring to, when we use the term stem cell, there's actually a bunch of different kinds of stem cells. Some are, pluripotent, some are totipotent, meaning that some cells, like embryonic stem cells, for example, can become essentially any cell in your body. Whereas, like a neural progenitor cell, which is sort of like a stem cell, can become any neuron. Well, theoretically, any neuron. And like that's sort of how it goes, right? It's sort of like a spectrum of just how versatile a cell is. And so my suspicion has always been that the reason you would use like a neural progenitor cell or some kind of somewhat differentiated stem cell is that what you want are dopamine neurons, right? Like you don't want skin cells, you don't want nails, you don't you don't want like teeth, in your brain. But who knows what those cells might be exposed to in any given context, and then maybe you do start growing, an eyeball in your ventral tegmental area or whatever. And so my guess is that it just eliminates a lot of that potential risk, the repertoire of potential differentiation to just, the ballpark of what you're trying to get. That would be my guess.
Right. But I think like epithelial stem cells are becoming ubiquitous in most applications, especially like in regenerative.
Oh, yeah. Yeah. Here, just this is an interesting topic, and I think it's just worth talking about. So, what Abyss here is talking about is that for a while, the only place that we could get stem cells from was embryos. at a certain phase where it's not like this, it's not particularly large. It's just this tiny little, like very soon after it forms, there's a little chamber within it that you can take out some embryonic stem cells. And again, those are like about as versatile as can be. But for a pretty long time, from the perspective of biomedical science, it was illegal in the United States to work with embryonic stem cells for obvious reasons. And what that did is it inspired a lot of really, really innovative research. And what happened was this group, I believe, what's his name? It's like Tamanaka or something, Japanese scientist, maybe Japanese American actually. I'm not sure, but he basically came up with a recipe to take skin cells and treat them with a bunch of enzymes, enzyme cocktail that cause them to essentially go back in time and de-differentiate into something like an embryonic stem cell. It's induced pluripotent stem cell, iPSC. Okay, so who cares? Why now that it's not illegal to do that, and it wasn't illegal in the entire world to work with embryonic stem cells, why do we care about this? Well, here's what's really, really useful about that. So let's say you have Parkinson's disease, and as we talked about, I think it was last week, got totally no track of time, but there is the prospect of curing Parkinson's disease by injecting stem cells into the certain part of the brain that's most directly affected by Parkinson's. So that's fabulous. Now, one of the issues with that kind of a medical treatment is that your body has immune system mechanisms that essentially exclusively evolved to recognize foreign entities, right? Foreign cells, bacteria, viruses, of course, but then also, foreign cells. And that is sometimes why pregnancy can be difficult, but it's also why transplants, like skin grafts and these types of things can fail because the immune system attacks it and damages it as though it were a virus or a bacterium. But if you can get you can take one of your personal skin cells, take a sample, and then turn them into stem cells, that eliminates a ton of that risk. And so therefore, it's almost like, it is literally like using cells from your own body to cure diseases in your body. And you avoid that immune system reaction. Interesting topic. So go ahead.
Yeah. And also, do you know if they used some kind of NGF, like neurogrowth factor? Because it's really difficult to create some kind of glial scaffolding, right? If you have stem cells, especially if it's gray matter ablation, because I believe that some kind of cortical degeneration would entail some kind of eradication of pyramidal cells in the gray area, so yeah, unless they use some kind of NGF or any kind of neurotrophic factor, I don't see that kind of binding to the brain as much as they hoped and providing the results that they expected. I just wanted to hear your opinion.
Yeah, I'm almost certain they did. I frankly cannot remember what they might have been, BDNF or NGF or any of these types of neurotrophic factors or chemotactic factors. What I would actually look at is the study in mice rather than see what they injected in that guy, that physician with Parkinson's, because I'm like 99% certain that it was like a cocktail, but I'm trying to remember. I think it was like surprisingly simple, or I honestly I can't remember. I'd have to look again.
Okay.
Yeah.
That's fine. And also, while you're discussing with Javier, it kind of reminded me that there are some molecular biologists that actually argue that the telomere argument for aging is kind of debatable, and they cite the very example that I just stated, which is like the epithelial cell into iPSC argument, because arguably skin cells have melanin wrapped around them that prevents further degeneration of the cells, but also their argument is that it's not the DNA that's actually doing the fragmenting, or even though the telomeres might shorten, the DNA may not be as damaged as much, it will be the sort of like the systemic effects that kind of stem from the cell differentiating, or at least like dividing over time, that can actually cause some kind of degeneration. But yeah, I just wanted to bring that to your attention if you heard that before.
Yeah. I mean, for sure, like at a minimum, I definitely know a lot of scientists at least argue that the like telomere theory of aging is an exaggeration. It's maybe a part of the story, but it's not the thing that regulates aging. Whereas it's sometimes portrayed to be to be that, particularly in the popular press. But yeah, no, I haven't seen the like argument derived from stem cells. That's interesting. Yeah, I mean, that's good. It's a good point.
I guess like my final question is just like an opinion for you. What do you think is like the ideal electrode design that's probably going to be implemented? That if we're talking about DBS using some kind of Neuralink style.
Dude, that is outside my wheelhouse, man. Like that, I never had to make electrodes. Like I made viruses, but yeah, it seemed like a big pain in the butt. But yeah. Sorry. I mean, but you know what? I think like 3 weeks ago, or who knows, whatever. Who cares? We were talking about one of the technologies that I think it can potentially be pretty transformative. It was the neural pixel. Are you familiar with this?
No, I'm not. What is it?
You should check it out. It's out of the Dieseroth lab, but it's basically. Oh no, it's not. It's not.
That guy.
it's not. I'm sorry. I'm wrong. He is using it though. He's using it. No, it's a guy named like Tim Harris or something. It's not Tim Ferriss. It's like Tim Harris, I think. Anyways, it's just like a single electrode that has like a ton of receptive components on it, and so rather than using a tetrode where you can like move it up and down basically in like almost a single plane, it's just like has a bunch of satellite dishes on its side, and so as a result you can get this very like deep and comprehensive slice of activity. And what the Dieseroth team did, of course, they went like over the top and they implanted like 40 of them or something like that into a mouse brain, and they like had essentially a readout of like global brain electrophysiological brain activity. Really, really remarkable. Just a totally different level of interrogation. But anyways, yeah, you should check it out. It'd probably be more interesting to you than it is to me.
Okay, will do.
Awesome, man. Well, thanks for calling in, dude.
No problem.
Bye. Bye. I know. It's bye. I don't know if you know that. 1,000 points to Gryffindor for anybody who knows who that is. Okay. I think I should call it a day. Have you ever wanted to eat monkey brain? No, I've never wanted to eat monkey brain. And in fact, I highly discourage anybody who wants to eat any kind of brain, just for even the prospect of being exposed to any kind of prion. Prions are nasty business. All right, I'll call it a day. So I have a YouTube channel. It's called youtube.com/anthropoid. Today I talked about the potential of metformin, a sort of frontline treatment for diabetes, type 2 diabetes, for any kind of cognitive benefits, specifically staving off cognitive deterioration or dementia. youtube.com/anthropoid. I have a podcast called Wired to be Weird. We talked about a lot of the topics we talked about today. We talked about in depth on the podcast. Wired to be Weird, iTunes, Stitcher, Google Play, etc. The longevity episode. If you're interested in these topics, the longevity episode, we talked about all of these topics. And I guess that's it. All right. Oh, thank you so much, Cosmic Crows. I appreciate that. Other than that, yeah, check out the YouTube channel. Subscribe if you are so inclined. And other than that, yeah, have a great week. See you next week. Same time, same place, right here. Be there. I'll see you there. Have a great week, everybody. All right, YouTube, thanks so much for watching, everybody. I really appreciate it. And let's see. Yeah. So if you're not subscribed and you're interested in these sort of free-form chats, or if you have any suggestions for topics to cover, I'm all eyeballs. So yeah. Other than that, have a great week, everybody. See you guys.